This podcast is brought to you by the Albany Public Library main branch and the generosity of listeners like you. What is a podcast? God, Daddy, these people talk as much as you do. Razib Khan's unsupervised learning.
Hey everybody, this is Razib, and I am here with my friend Dr. Abdel Abdellaoui. I think I like to call him Dr. Abi, but maybe you don't need to call him that. So Abdel, could you introduce yourself?
Yes, I'm Abdel. Thanks for having me, Razib. I am a behavioral geneticist based in Amsterdam. I do research at the Amsterdam University Medical Centers. And my research focuses on the behavior genetics, a bit of population genetics, a bit of psychiatric genetics. And I'm interested in human genetics in a very broad sense.
Yeah, and so I mean, how did you get interested in human genetics in a very broad sense? I would like to be curious about your background and just what the system for this sort of career path in the Netherlands, because I think you were educated in the Netherlands, like what that is.
Yeah, so first I started informatics, just computers, and then after a couple of years of learning how to program and work with computers, that it just became too detailed for me and a bit boring. And I found that I was more interested in humans, so I switched to psychology, which was kind of a risk professionally, because there was a lot of work at that time when I started in the IT business. But I just decided to follow my curiosity and just see where it takes me. So I did psychology. And then I quickly realized that I found the things that I was learning about the brain very interesting. So I did a master's in neuroscience. And then within that master's, there was a course about genetics. And that got me really fascinated. So I zoomed in. I just kept zooming in, and then until I couldn't go any further. And that's DNA. And and I was hired also to work part time at the department that was giving those genetics courses. So and I enjoyed myself very much there. I quickly made friends, and then I got offered a PhD position, and everything fell into the right place, it seems. And
I gotta ask. Have you been within the same institutional framework then your whole career, or since you started your education?
No, not my whole career. So I did my PhD there, and then like during my master's, I went for a half a year to Brisbane, Australia, and then I did my PhD here at that same institution. And following that, I did a postdoc, and then I went to Australia again for a very short time, a couple of months, and then I switched institutions. So I'm now not anymore at the same institution, but we are the place that I work now does consist of people who all started there. We are sort of a spin-off of that institution. So the institution where I did my PhD, that's the Netherlands Twin Register. That was became famous for the gigantic data set of twins since like the end of the 80s. They invited every twin that was born in the Netherlands gets this gift basket, and that includes a brochure of the Netherlands Twin Register. So every twin that is born, almost everyone is registered and gets questionnaires every couple of years, and some come by for things like brain scans, and that was a great resource to do genetics research and a lot of twin studies, which is what I started doing. Yeah, so that was that is sort of a hub here in Amsterdam for a lot of the geneticists that come from here start there, and some of them have started their own lab. One of the spin-off labs is where I'm working now.
So, I mean, was this registry started for genetic research or psychological research?
Yeah, for genetics research. Yeah, as far as I know, it is at the psychology department. It's called biological psychiatry, psychology. So yeah, mainly behavioral genetics, but it's yeah, those genetics methods you can apply the ones that you can apply to behavior. You can also apply them to physical and mental health. So it's that's also what I like about the field, is how interdisciplinary it is, how easy it is to study the whole human. And if you have broad interests like I have, then it's a very good place to be.
Well, I mean, let's talk about your interest. So you are very active on social media. Anyone who has not followed you and is on Twitter and is interested in topics that we're going to talk about should follow you on social media. Doctor underscore APPIE, Dr. Abi. So you tweet a lot about behavior genetics, which is a distinct field with a particular intellectual tradition. But I think you became pretty well known a couple of years ago for a paper on stratification of I think like intelligence and income in Britain, and to not beat around the bush, like people made accusations you guys were doing eugenics and all of this stuff. So I guess the question is twofold. How did you get interested in that particular topic? And can you just talk about the general results? And then second, did you even expect that sort of reaction? Like, did you know what you were going into?
Yeah. Um, yes. How did I get into that topic? I'll start with that. When I first got my hand on genetics data, it's those they're huge data sets, and there's so many measurements, so many genetic variants, millions of them, and it's hard to make sense of them. And I was interested in just first looking at the largest patterns of genetic variation. So that's what I did before that study that you mentioned, and I did that with a Dutch data set, and I saw that the large patterns of genetic variation reflect ancestry differences, and I also combined that with geography. So if you plot those large patterns of genetic variation on a map of a country, of the Netherlands or of the Britain, then you get very nice geographic patterns. So I did a couple of articles on that, on how those patterns emerge, how they are maintained, and then I got my hand on a British data set, the UK Biobank data set, which consists of 500,000 individuals, which is a lot larger than the Dutch data set that I was working with. That was about 10,000 individuals. So I could now ask questions, what's beyond those large patterns of ancestry, and look at the geographic distribution of genes that are associated with heritable traits. So how are the genetic predisposition for heritable traits, how do those look if you put them on a geographic map after you control for those ancestry differences that I studied before that? Yeah, so I looked at a wide range of traits: physical and mental health and personality, and anthropomorphic traits, things like height and BMI, but also educational attainment and cognition. And I just, without knowing what I would be finding, I just looked at how strongly the genetic predispositions for those traits cluster geographically. So which ones show the largest regional differences? And by far, like it was an outlier trait compared to the rest, was educational attainment. So genes associated with educational attainment showed the strongest geographic clustering. And when you plot that on a map, then the distribution looks a lot like the differences between the richer regions of the country and the countries that are struggling economically. So so as part of that same article, I tried to figure out how these regional differences came to exist. And we saw that people that migrate out of those poorer regions of the country, they have a higher genetic predisposition for a higher educational attainment than the entire country on average. So the people that are able to move out of the poorer regions, those are the people with the highest educations, or at least the genes that are associated with that. And that kept increasing those regional differences throughout the century.
What I was saying is, I think the reason that your paper elicited such a strong reaction is because there's all this stuff related to the north of England and the south of England in the context of English culture, and you just made a lot of comments about that as an outsider. You kind of stepped into a big cultural debate.
Yeah. So to answer your previous question of why I was interested in this, so I this was not something that I intended to write about. It was just something I found because of my approach of looking at what are the biggest patterns of variation in the data. So this is what I ran into, and of course I realized that it was a sensitive topic. So we were very careful in, first trying to find out, is this really real, what we're finding? So we spent a lot of time trying to figure out whether those signals were real, and also thinking about how we were going to communicate it. So we spent time and energy on trying to explain very clearly what our results meant, and also what they didn't mean. And I think that helped pretty well. The journalists that I spoke to, I think they did a great job at reporting it with the necessary nuances of it, which I didn't really get into yet in my answer to you, because it's not the complete story that there are genetic differences. Like those genetic differences, they coincide with a lot of environmental factors that are associated with living in a poor neighborhood versus living in a rich neighborhood. And that makes those genetic effects that we see correlate with all those environmental factors. So if you grow up in a poor region, you are likely to have a lot of cards stacked against you, in also just in an environmental sense. There's much more stress in those neighborhoods. There are way more fast food restaurants, for example, and we also see that there are higher diabetes rates and obesity rates, and those kinds of health differences are not as strongly associated with the genetic differences. So those environmental factors, they have an effect on people's health, but also on things like their educational outcomes and cognition. So we also tried to make clear that it's very difficult to tease apart the genetic effects and the environmental factors. And that's also part of the reason why people are moving out and why those regional differences are increasing, is because it's very hard to live in those neighborhoods, and if you can get out, then you do.
Yeah. Well, I mean, so you're bringing all these complex variables into play in your discussion, but people were not focused on that, right? They're focused more on like, oh my God, he's talking about genes and stuff like that, and he's talking about eugenics. Like, how surprised were you by that reaction?
Well, to be honest, I didn't see that many of those reactions. Like there were some people which I expected that had those kind of reactions, but the vast majority was that I saw. they did take that to read the nuanced reports and the summaries of the papers that we provided, and I wrote like 3 columns about it to summarize what we found. And overall, I found the reactions to be quite positive. I did when we posted the preprint a year earlier, we got a lot more negative reactions to that, and that might also have to do with like the surprise factor, and we just dropped it online and we didn't really provide like a frequently asked questions document and columns and all the talking to the journalists that accompanied the actual peer-reviewed publication. Yeah, but so we've learned from that, obviously, but from the actual peer-reviewed publication, which got a lot more attention also in the media, but also just on social media, I think those reactions, at least what the parts, at least what I saw, was positive for the largest part. Well, positive. People thought it was interesting, and there wasn't a lot of the pushback that apparently you saw.
Yeah, well, maybe I'm following the wrong people. Maybe that's what you're saying. So, I mean, this whole area of behavior genetics, genomics, it's really blown up in the 21st century. Like what do you think about its past history and the way people view it because of that? Because some of the reactions that I'm talking about are just due to what people perceive as the misuse of twin studies, twin registries, the controversy around Cyril Burt, the famous twin researcher, and his possible fraud. Like all of this history, like do you think that you're coming into it fresh in the 21st century, or do you feel like the field and possibly you yourself would have to address these sorts of questions?
Yeah, no, I absolutely think that it's important for people to be aware of the past, and that it did have a nasty past, and it's good that people are critical and that they ask tough questions and try to make sure that we don't repeat our past mistakes. And people that are new to the field should also be educated on that past. I totally think that's justified. Yeah.
So basically, you're saying people should be doing their thing, just like offer the criticisms that they feel they should offer, but probably just have them on solid ground. So with this sort of behavior genetic research, there's all these environmental confounds. Uh, what you've been talking about, alluding to. Can you talk about like just in terms of technically how you can partition genetic and environmental effects, and just like how confident you are about that sort of thing? Because I think one of the issues that people have is these publications are published, they're out of the UK Twin Biobank. How generalizable are they? I mean, do they have any relevance? Is just another line on a CV? Can you just address those
Yeah.
questions?
Yeah. So those are things that we are trying to deal with now. It's becoming more and more clear how intertwined those environmental and genetic effects are. It's on a regional level, as I explained earlier, but also on a family level. For example, if you have parents with a higher education, you inherit their genes, but you also inherit the environment that they are able to provide for you because they have more resources, because they are higher educated. So that sort of creates this gene environment correlation that makes it difficult for us to estimate exactly how strong those genetic effects are. So there are several ways in which we are trying to deal with that now. People are looking at within family effects, so just comparing siblings with each other who are just slightly different genetically, but they are raised in the same environment. So that makes it possible to tease out partly those genetic effects, but not the genetics from the environment. I'm also working on a study now where we try to do the same thing, but on a regional level. So we try to do, to estimate these genetic effects, accounting for which regions people live in. And we already see that that has an effect on the genetic signal, especially those genes that are associated with educational attainment, because those are most strongly associated with where you get to live. So if you estimate the genetic effects of BMI on BMI, for example, and you account for where people live, then that removes genetic effects that are associated with educational attainment from that BMI signal. Yeah, so those are types of approaches that we're using now to deal with that, but that's all work in progress. But yeah.
Mhm. Well, I mean, so how has genomics transformed behavior genetics? Because obviously, before people knew what relatedness was as an expected value, so siblings are related zero point 0.5, but now you can figure out like how many IBD segments, like, how, if they're 47% related or
Yeah.
52% related. Like, how has that changed your work? I mean, I'm assuming it's added a little more precision and power, but could you talk to that?
Yeah, so it made it easier, as I just said, to tease out environmental effects from genetic effects by comparing siblings with each other, by being able to more precisely estimate how genetically different they are. And of course, genomics makes it able to compared to twin studies to pinpoint genetic regions that are associated with the traits. So
So yeah, you can return. So you can return a specific gene for some sort of like liability
yeah,
or
yeah,
characteristic,
yeah.
right?
So I'm hesitant to be all too positive about that, because it's very difficult to those effects. We also, like, it made us realize how small the individual genetic effects are, so we are able to pinpoint genetic variants and say this genetic variant is definitely associated with the trait, but the effect is so small that it's difficult to go a lot further with these individual genetic variants. There are a small number of examples where they did lead to significant biological insights. For schizophrenia, there is one specifically strong signal on chromosome 6 that they have now figured out that it influences some type of synaptic pruning that's especially active during adolescence. So that was an important biological insight, but such insights are quite rare, especially in behavioral genetics. But in aggregate, if you take all those individual genetic effects and you add them up, then suddenly they start explaining substantial amount of variation, and you can use those the aggregate of those genetic effects to build something called polygenic scores, and that makes it able to
Mm-hmm.
predict someone's genetic propensity for a certain trait in an independent sample. So that's something that's only been possible since this
So
genomics were.
I do have a question related to that. So you do a lot of psychiatric genetics and whatnot. So schizophrenia, autism, these are highly heritable traits. I think they're like on the order of like zero point 0.7, zero point 0.8, 70-80% heritable. You can correct me if I'm wrong on that. But they are still polygenic. They're still due to different genes. Can you talk about the genetic architecture of different cognitive behavioral characteristics in terms of, are there some cases where it looks like there are larger effect alleles or loci due to possibly mutation, along with the common variants, and in other cases, just mostly common variants. So, for example, intelligence is, I think you just say it's common variants, right? But what about schizophrenia and autism? Is it heterogeneous, where there's some cases there are large effect variants in some people, and other people
Yeah,
no?
yeah. So I wasn't for schizophrenia and autism. I wasn't involved in those genetic studies, but I do use their results for a lot of the follow-up studies for a lot of the stuff I do. So I do know that it's common variants play a very big role. in all those psychiatric disorders. But in general, the rarer variants individually seem to have larger effects. So the common variants, they don't explain all of the heritability that is expected from twin studies. And the expectation is that a lot of the remaining heritability is due to these rare variants that individually have larger effects. They've showed this to be the case for height. So for height, they now with a huge study of 4 million participants. I think they just that this study is not out yet, but it's been it was presented recently at some conference that they recovered almost all of the heritability using 4 million participants, and that the remaining heritability that they didn't find before, that was all due to rare variants. And the expectation is that the same thing is going on for psychiatric disorders. Maybe even a bit, yeah.
So was this the Vischer
Yes,
Group's
exactly.
presentation? Okay, yeah, I've been present when he presented that, actually, so I know what you're talking about. He's presented in multiple different places. So basically, to recap for the listener, I think what they did here is they took whole genome sequences with large human populations, correct?
Yeah, I think so.
Or was it SNP arrays?
No.
Yeah, okay. I think it was whole genome sequence. Some of it was whole genome sequences. How do you find rare variants?
Some of it was whole genome sequence. I don't remember the specifics, but there was definitely whole genome sequence involved.
So I think there might be extension on some of the work that I've seen Peter Vischer present. So if it's 4 million individuals, it's probably not all whole genome sequences. So it's probably a joint effect of whole genome sequences with SNP arrays. So whole genomes would be all 3 billion base pairs. SNP arrays would be hundreds and hundreds of thousands, possibly millions of the base pairs. And so to get rare variants that are unique, you would want whole genome sequences, whereas the SNP arrays would catch all the common variants. And so I think what you're describing here is new work using joint effect of common and rare variants using these two methods.
So there was a preprint before the 4 million study where they used only whole genome sequencing, and there they already could see that for both BMI and height, that a lot of the remaining heritability was because of rare variants. And I think for the 4 million study that was just a GWAS where they could actually have the whole SNP heritability match the twin heritability, if I'm not mistaken.
Yeah, and so what you're alluding to here is you use classical methods, statistical inference type stuff with twin studies where you calculate the heritability by comparing the differences between normal siblings and identical twins. And, what's the correlation here between these sibling pairs? And then also now looking at genetics and genomics, and there's traditionally been a gap of the missing heritability that started about a little over 10 years ago. And one of the hypotheses of the gap is we just didn't have the technology to catch all the variants. That looks to be correct.
Yeah, there was a lot of mystery surrounding the missing heritability back in the days. There were a lot of different explanations. A lot of people started to doubt the whole twin design and believing that we were wasting our times measuring those SNPs because the assumptions of the twin studies were wrong. Some believed that we were wrong to measure SNPs at all. We should look at structural variants, which are often correlated with the SNP variants. But it turned out well, first of all, that the genetic effects are individually just too small to detect with the sample sizes that were available back then. That was the largest part of the missing heritability, and all of the remaining stuff, at least for height and BMI, probably also for psychiatric disorders, is those rare variants.
So I have a question about papers and preprints. So you have a new, I think it's a preprint out with David Hugh Jones, Natural Selection in Contemporary Humans linked to Income and Substitution Effects, with 400,000 British subjects with European ancestry. Could you talk a little bit about what you found in that preprint?
Yeah, so it was replicating, I think there were 4 or 5 studies that looked at, before ours, that looked at contemporary natural selection, so natural selection that's happening now, by looking at the number of offspring that people have. And they all report that the strongest correlation was with educational attainment. So genetic effects that are associated with educational attainment are associated with the number of offspring, and that correlation is negative. So people with a lower genetic predisposition for educational attainment have more children. So David and I looked in more detail by splitting up the sample. Trying to look at how that works, what the mechanism is behind it. And what we see is that there are actually 2 opposing forces seem to be at play here. The most striking thing I saw therein was that if you split the sample into, if you look at the age that people start to have children, then you see that if you look at mothers that have their first child at the age of 22 or later, then suddenly that association is reversed. Then people with a higher education have more children, and so the overall association seemed to be driven by people that have children at a very young age, so before they were adults, often, mostly, if you define adult as 21 years old. So there are opposing selection pressures, if you will, and David Hugh Jones, who is an economist, he tried to put it in he tried to put it in an economic theoretical context, and there's some theories that were from the 60s or 70s that predicted there's a substitution effect and an income effect, where people with a higher income are able to afford to have more kids, but if you have kids at an early age, then you also give up some of the potential income that you could gain in a job market,
that's called an opportunity cost. I think in economics, you got some trade-offs here. So one reason you're studying the British data is because you have it, and then you're talking about the Netherlands Twin research. So a lot of your research seems to be due to where's the data, which makes sense. What is your anticipate? I mean, are there other data sets coming along that you're interested in? I mean, I know the Nordic countries, they often have registries and data sets. And then in terms of the possibilities international, when can we get more genuine insight into stuff going on in non-European countries?
Yeah, so that's something that's very lacking in genetics research nowadays. I think almost 90% of all the genetic data is in the studies that are published are from countries with a European or participants with a European descent. People with non-European descent that are included in those studies are usually excluded for methodologically good reasons, because it brings a type of confounding to the signal called population stratification, where the systematic allele frequency differences between ancestries makes it difficult to estimate genetic effects. But that makes the results very based on very homogeneous data sets, and so and it turns out that a lot of those findings are hard to generalize to non-European populations. The polygenic scores I talked about, they work way worse if you compute them for non-European populations for a variety of reasons. But the field does realize that.
Abdellaoui, what are those reasons, just so the listener knows?
Well, part of it is differences in environmental factors and cultural factors that make phenotypes, those traits systematically differ between populations, and those environments can correlate with genetics in their own way in different cultures, in different environments, different geographic regions, and also the way that genetic variants correlate with each other, something called linkage disequilibrium, because we don't pass down separate genetic variants to our offspring. We pass down whole segments of DNA to our offspring, and that makes genetic variants that are closer to each other more correlated with each other. And those patterns of correlations also differ between populations, and they affect the genetic effect that is estimated in a genetic study, because most of those studies are not based on sequence data where you measure literally every variant. They choose specific genetic variant based on the correlation with other variants around them, so that they can effectively capture a lot more genetic variation while only measuring an affordable amount of genetic variants. But if you do that, those DNA chips that select those genetic variants are based on European reference data sets, and so that influences the signal, and it makes it difficult to translate to other populations.
And so, I mean, what are you looking at in the future? I mean, this is like going to be another 10 years, the 2020s. We have all these computing tools and techniques and data sets. Like, what are you super excited about in terms of traits, data sets, questions that you're exploring?
Well, I hope that people will be investing, and it looks like they probably will, in more diverse data sets and also more family data sets, so that you can try to tease out the environmental effects from the genetic effects. Also try to get more geographic coverage, because we now see that those regional effects also make it more difficult to reliably estimate genetic effects. So just bigger data sets. So like the field has shown that it's capable of reaching. It's a
Yeah,
really
the bigger your sample size, the more power you get,
yes, yeah.
basically. Like in terms of
The bigger
So
the
I
data
mean,
set, the
I think like
better.
one of
But
the
also, if you don't, if you have a systematic, what we call ascertainment bias, if you don't sample well enough, then all those confounding signals will also get stronger if your data set grows. So it's not just a matter of sampling more people, but also thinking good about how you sample them. So I hope that they will include more family members and better geographic coverage, and also more non-European people. If they do that, then that would be a good direction to move to.
Yeah, I mean, I think one of the historical contexts that we need to recall is when the first genetic, kind of like linkage studies, that sort of thing, were done in behavior genetics. It produced a lot of false positives that weren't validated, and that's just because they didn't have the power.
Yeah.
It was 20 years
Mhm.
ago. Now we have the power to capture these polygenic traits, and it's just it's a whole new world. So you did mention like non-European populations. One of the questions that I have is like getting the phenotypic information in these societies. So I mean, are there projects that you know of that are starting up to survey and do longitudinal tracking over the decades in non-European
Yeah, well,
societies?
I know there are some Asian countries that are collecting very large data sets that I'm excited about. In China, they have sort of a UK Biobank like thing going on. In Japan, there was a project called 100,000 African Genomes Project or something. I might have gotten the name wrong, but that sounded like something to be excited about. So there are definitely projects going on, and we realize in the field that it's a problem that we are so Eurocentric. And I do expect to be able to study more diverse data sets. I hope that they will also follow the example of UK Biobank of making the data sets public, because that was very helpful. I think that at the moment about 15,000 scientists or something like that, might have been more by now, are working on the UK Biobank dataset, and that's really great.
So you've been a little fire hose of publications recently. What are you most excited by in terms of a result or a paper that you're working on or a project right now? Like, as we close out the conversation, I'm curious what the big thing that we want to hear from you. And, again, follow him on Twitter, Dr. underscore APPIE. It's really interesting stuff. All of this, discussion we're having, like 10 times more intense, when he can link to things and comment.
Thanks for that, Razib. Yeah, what I'm excited about now. Yeah, so we're working on several projects simultaneously that I'm excited about. There are some interesting phenotypes that we are focusing on. I don't want to give those away yet to prevent us from being scooped. But I think those within the next couple of months we should have those. I don't do that often. I don't often focus on specific phenotypes. I try to keep it as broad as possible. But we do have one or two interesting phenotypes coming up. And I'm working on two sort of overview / perspectives papers where I try to put all of the findings, the very recent findings, some of them only out in preprint, some of them not published yet, to try to put those in a larger context. I've been working on those for the last one of them for the last year. That's now the revision has just been submitted, and another one I'm just finalizing. So I'm excited about those, because I don't do those often either. I don't really do review or perspectives papers. Usually I present new results. So those, yeah.
I would say reviews, it's one of those things where it's not as prestigious or interesting for the researcher, but it's incredibly useful for people outside of the community.
Yeah, and I really enjoyed writing it too. It was also good for me to get to put all those things, in one story and one, and it's not just reviewing, but also like some interpretation that's involved in that. So that, I enjoyed that very much. So I'm looking forward to sharing that hopefully soon as well.
All right. I think, we touched the main topics that I want to talk about with you, Dr. Abdel Abdellaoui. So, I think like people are going to be hearing from you, seeing from you. So, it's been great just talking about your research and how you got there, and, just all the resources that you have. I think that's the big thing that people need to take away from this. There's a lot of resources out there, and you're just taking advantage of them, right?
Yes, yeah, I'm so lucky to have started in a time where these resources were being made available to the public. Without them,
Yeah.
I don't know what I would have been doing, honestly. Yeah,
Probably being a code monkey
yeah,
somewhere, right?
probably.
Yeah. Compiling some code. All right. It was great talking to you,
Yeah, great
Abi.
talking to you too. Thanks for having me.
This podcast for kids